Hello everyone, this presentation is about a major trait of small ruminant resilience, resistance to gastrointestinal parasitism, and in this study we use divergent line to investigate if selection for resistance in young sheep was also effective in the adult female, and we also looked at trade-off with production traits. So this work was done in collaboration between our team in Toulouse with a Scottish partner from the Roslin and the Moreno-Romieux. Gastrointestinal parasitism depends on pasture, so it's a major hindrance to the development of sustainable grazing systems. Moreover, the main option that was to use chemical control is currently compromised by the emergence of drug-resistant parasites. So this map, for instance, in France, shows that there is an increasing number of resistance to the main molecules that are used in the field. So we know also that there is genetic variation in fecal egg counts, and we can use these traits to select for resistance. And there is increasing prospect for this solution as an alternative to chemical control. To select for resistance, we can use fecal egg count based on natural infections. However, it's not the most accurate option because there is high variation in fecal egg count due to the parasite diversity, for instance, or the parasitic load. Otherwise, we use artificial infections with a specific parasite and with a controlled number of parasites that is ingested by the sheep. And we showed that this strategy was actually effective to select for resistant rams because their response to infection in control centers was highly correlated to the response of their progeny on farm. Finally, we can also use artificial infection as an experimental mean to study resistance to parasite, and this is what we've done in this study. We investigated two questions. The first was to see if selection for resistance in lambs is also effective in the adult female. In particular, around lambing, we know that immunity can be relaxed, and this may depend on the protein level in the diet. And the second question was to investigate the presence of trade-off between resistance to parasites and production traits like growth and maternal condition. For this study, we used our divergent lines. These lines, they were created based on more than 250 animals, and after 2 generations, we obtained 53 resistant females and 44 susceptible ones. This divergent selection was based on a unique phenotyping protocol that included 2 artificial infections. The first infection was to initiate the immune response in the graying lamb, and it lasted 3 weeks, after which we had a 2 weeks recovery period, and then a 5 weeks infection based on 10,000 larvae. After each infection, we measured the fecal egg count, and we calculated a genetic index of resistance. And this index was divergent enough between the two lines. We had a divergence greater than 3.5 standard genetic variation, so enough to start an experimental design. Our experimental design included 4 different infection protocols. The 91 lambs of the second generation, all females, were first involved in the first infection protocol that was exactly the same as the protocol used in the divergent selection, so 2 infections separated by a 2 weeks recovery. And during this phase, the graying lamb were fed concentrate ad libitum. After this, mating occurred, and the ewes with at least 2 lambs were involved in the second infection around peripartum. This was a trickle infection, so they received about 1,000 parasites every week for 8 weeks. Half of the group was fed high-level protein, and the other half was underfed. After this, we had a second mating, and we had a third infection protocol in early pregnancy with 1 single dose of 10,000 parasites. Ewes were fed with a regular diet. And finally, we had another infection in peripartum with the ewes with at least 2 lambs. And contrarily to the first infection in peripartum, we had an infection with 1 single dose, and ewes received high level protein during this infection. we did many measurements, but here we will focus on two main types. The first type is the response to infection with the fecal egg count and the hematocrit loss. So the hematocrit loss is the variation compared to the initial level before infection. And it's associated with the loss in red blood cells with this parasite, Haemophilus contortus. The second type of threats is the production threats of the U with the body weight and the back fat thickness and muscle thickness measured by ultrasound. Now let's have a look at the results. So the fecal egg count for the 4 different phases of infection. As you can see, we observed a large difference between the lines in the LEM phase, and this was expected. It's a direct response to selection. But we also observed the line effect most of the time in the reproducing U. However, this effect was lower around lambing in the first peripartum with trickle infection, and to a lower extent in peripartum 2 with the single-dose infection. The effect of protein was non-significant, and the response also were consistent in terms of hematocrit. We observed a loss in hematocrit that was higher in the susceptible line compared to the resistant line. Here are the phenotypic correlations between the measures observed in the different stages, and we analyze separately pregnancy and lactation in the 2 peripartums. So for fecal egg count, as you can see for the correlation between the LEM phase and the adult phases in this first column, the correlations were higher than 0.5, so quite high, and they were in the range of the values that were observed for the correlation between the adult phases. In contrast, for hematocrit loss, we observed that only one value for the LEM phase was significant with the measurements observed in the adult phase. So this confirms that fecal egg count is a better measure of resistance than hematocrit loss. Then we looked at potential effect of resistance on new weight and condition, so a trade-off between production traits and resistance. And for body weight, we did not observe any effect from the line, either in the LEM phase, peripartum 1, early pregnancy, or peripartum 2. In the peripartum 1, we did observe an effect of the diet on the body weight. However, this effect was not different between lines. In terms of back fat thickness, we observed a potential trade-off between back fat deposition and resistance, because the resistant line has a lower back fat deposition compared to the susceptible one. However, this effect was not confirmed in the adult stages. In contrast, for muscle thickness, we did not observe any effect of the line in the LEM phase. However, there was a trend for the resistant line to have a higher muscle deposition compared to the susceptible. In summary, we observed that selection for resistance in Graham lambs was also effective in the adult breeding views most of the time, except in peripartum. We also observed that the level of protein in the diet had an effect on ewe growth and also on the lamb growth. This was not shown, but this level of protein did not affect the ewe resistance to parasites. And thirdly, we did not observe much evidence for trade-off between resistance and production traits. There was some evidence for trade-off during growth, where we observed that the resistant line had a reduced fat deposition compared to the susceptible line, but there was not much evidence for trade-off during the reproductive phases. And this lack of evidence was confirmed with multivariate models using repeated measures. So now there are 2 ongoing studies related to this work. One is to better understand the relaxation of immunity around lambing and the potential role of protein allocation. So for this, we used protein labeling based on a heavy water injection. And the second perspective relates to the use of data for modeling. So for this, we built a mechanistic model that described the trade-off between back fat thickness and resistance. And with this model, we will explore the consequences of selection for resistance at the farm scale. So you can have more details on this part in a poster that is presented in the current session. Thank you for your attention.